In this section, we will talk about what is glomerular filtration. Do you know what is glomerular filtration? Well, glomerular filtration is the process by which the blood is filtered, while passing through the glomerular capillaries by filtration membrane. It is the first process of urine formation. The structure of filtration membrane is well suited for filtration. Now, do you know what is filtration membrane? Let's discuss it. Filtration membrane is formed by 3 layers. First is glomerular capillary membrane, second is basement membrane, and third is visceral layer of Bowman capsule. First is glomerular capillary membrane. Glomerular capillary membrane is formed by single layer of endothelial cells, which are attached to the basement membrane. The capillary membrane has many pores called as fenestrae or filtration pores with a diameter of 0.1 micron. Moving towards the basement membrane. Basement membrane of glomerular capillaries and the basement membrane of visceral layer of Bowman capsule fuse together. The fused basement membrane separates the endothelium of glomerular capillary and the epithelium of visceral layer of Bowman capsule. Third layer is visceral layer of Bowman capsule. This layer is formed by a single layer of flattened epithelial cells resting on a basement membrane. Each cell is connected with the basement membrane by cytoplasmic extensions called as pedicles or feet. Epithelial cells with pedicles are called as podocytes. Pedicles interdigitate, leaving small cleft-like spaces in between. The cleft-like space is called as slit pore or filtration slit. Filtration takes place through these slit pores. This side has blood, while this side has filtrate. Now, another question arises that what is glomerular filtrate and ultrafiltration? Let's discuss both one by one. In process of glomerular filtration, when blood passes through glomerular capillaries, the plasma is filtered into the Bowman capsule. All the substances of plasma are filtered except the plasma proteins. The filtered fluid is called as glomerular filtrate. Now let's discuss the second term called as ultrafiltration. Glomerular filtration is called as ultrafiltration because even the minute particles are filtered. But the plasma proteins are not filtered due to their large molecular size. The protein molecules are larger than the slit pores present in the endothelium of capillaries. Thus, the glomerular filtrate contains all the substances present in plasma except the plasma proteins. There are some other few important terms that are needed to be clear, so let's discuss them. First is glomerular filtration rate. Glomerular filtration rate, or GFR, is defined as the total quantity of filtrate formed in all the nephrons of both the kidneys in the given unit of time. Normal GFR is 125 milliliter per minute, or about 180 liter per day. Another term is the filtration fraction. Filtration fraction is the fraction or portion of the renal plasma which becomes the filtrate. It is the ratio between renal plasma flow and glomerular filtration rate. It is expressed in percentage. Filtration fraction is equal to glomerular filtration rate divided by renal plasma flow into 100. Glomerular filtration rate is 125 milliliter per minute, and renal plasma flow is 650 milliliter per minute, which is multiplied by 100. These two units are same, so they cancel each other. 125 divided by 650 equals to 0.192 into 100. So the answer is 19.2%. Normal filtration fraction varies from 15 to 20%. Moving towards the method of collection of glomerular filtrate. Glomerular filtrate is collected in experimental animals by micropuncture technique. This technique involves insertion of a micropipette into the Bowman capsule and aspiration of filtrate. In 1924, Urine and Richards published one of the most important single contributions to renal physiology, as Gottschalk noted in 1992. Urine and Richards had developed and applied the method of micropuncture in frogs. The work provided the first experimental evidence that macromolecules, like proteins, were excluded from the glomerular fluid in Bowman's space. Moreover, they reported that concentrations of glucose, chloride, potassium, urea, and pH in glomerular fluid were essentially identical to the aqueous phase of plasma. These findings supported the concept proposed earlier by Ludwig, that tubular fluid is formed by a process of ultrafiltration across a capillary in which large proteins excluded. Urine and Richards also provided first evidence for tubular reabsorption in as much as the bladder urine was free of sodium chloride and glucose. This was all about this section. In our next section, we will talk about the pressure determining filtration.